Hello, good afternoon, everybody, and thank you very much indeed for tuning in to the 2015 CNEN Virtual Symposium. I'm Kevin Davis, the publisher of CNEN, and it's my great pleasure to introduce our closing speaker after a full day of great talks here, Nancy Kelley. This talk will be a slight departure from the dozen or so talks that you've heard during the course of today. Nancy is not a chemist. She's a lawyer and an economist by training. But she really is somebody who's truly made a major mark in the biosciences field. She led commercial real estate development in life sciences for many years, finding homes for multiple biotechs in and around Boston, and leading the development of the East River Science Park in New York. She trumped that, no pun intended, by conceiving and spearheading the creation of the New York Genome Center in Manhattan a few years ago. A truly remarkable feat, not just for raising $100 million in initial funding, but also, and I think even more impressively, for forging a truly unprecedented alliance among all of the leading New York academic institutions to get that center off the ground, and it's really flourishing. Over the past couple of years, Nancy has turned her sights to a new area, the exciting field of synthetic biology. And today she'll present some of the scientific and social opportunities that synthetic biology represents and discuss the logistic and funding models that are probably going to be needed to realize those ambitious goals. Her talk is entitled Engineering Biology for a Sustainable World, and it's my pleasure to welcome now Nancy Kelley.
And thank you for having me here this afternoon. I'm very excited to be here and to be sharing on a topic that I have learned a lot about over the last couple of years and which I think has great promise for the world. So as you said, the title is Engineering Biology for a Sustainable World. And this really came about through the Human Genome Project, which was one of the great feats of exploration in history. It was an inward journey of discovery through an international research effort to sequence and map all of the genes of human beings, together known as the genome. Completed in April 2003, the Human Genome Project gave us the ability for the first time to read nature's complete genetic blueprint for building a human individual. Those genetic instructions were contained in a molecule called DNA. And there are 4 basic building blocks, as most of us know, called nucleotide bases that are found in DNA. Adenine, cytosine, guanine, and thymine. It is the sequence of these nucleotide bases that encodes biological information. The cost of sequencing that one genome was about $3 billion. It took 13 years to accomplish. And today, just over 10 years later, that cost sits at about $1,000 and can be accomplished in less than a day, which is truly extraordinary development in only the 10 or so years since the Human Genome Project was completed. Now, due to the advances in the cost and speed of sequencing DNA, we've learned how to read the genetic alphabet A, C, G, T, and see how those nucleotides code for functions within cells. This has resulted in tremendous insight in using genetic information for improved healthcare delivery, where patients today get genetic screenings and personalized treatment for a number of diseases. Now we can read, but we're just learning how to write. The next century will be about how we write DNA using those nucleobases or basic building blocks to design and build bespoke organisms that can provide solutions to many of the planet's most pressing global challenges. You might be tempted to think this already exists as biotechnology. Biotechnology, after all, harnesses cellular and biomolecular processes to develop technologies and products that help improve our lives and the health of our planet. We have used the biological processes of microorganisms for more than 6,000 years to make useful food products such as bread and cheese, and to preserve dairy products. So what has our ability to read and write DNA allowed us to do that is fundamentally different now? Scientists now envision using those 4 basic building blocks of DNA to design entire biological systems from the ground up with as much precision as an engineer can design a bridge. Think about it. DNA and its basic building blocks, A, G, C, T, hold the genetic code for every function within an organism. Just like infinite permutations of zeros and ones form the basis for designing and writing software code that drive all functions of technology, these nucleobases can be used to write the genetic code for and build entirely new biological systems from the ground up. The practice is called synthetic biology or engineering biology in the most recent vernacular. The coming revolution in engineering biology will transform the industries of energy, agriculture, healthcare, chemicals, and bioremediation, to name just a few. And it will also raise social and ethical questions, including what constitutes life and what it means to be human, as well as important security concerns about biosafety and bioterrorism. Synthetic biology bridges a number of scientific disciplines, such as metabolic engineering, molecular biology, microbiology, systems biology, synthetic chemistry, chemical engineering, and genetic engineering. It also relies on a constellation of supporting technologies like DNA sequencing and synthesis, laboratory automation, design and modeling software, bioinformatics, and big data and computing. In theory, how does all this work together? To make biology engineerable, scientists must construct predictable systems and standardized parts. To do this, they use computer software to write the code that design organisms in the abstract and predict their production. Ideally, you want to design, build, test, and scale up your organism in a predictable, modifiable way. The first part, constructing predictable systems, is done by understanding how complex organisms like E. coli or yeast work and stripping away as much DNA as possible, leaving only the most rudimentary functions. The organism is now just a foundation, a platform for new DNA. The goal is to create a platform with well-known characteristics and functions so new genes perform their intended role without being disrupted by other cellular functions. Once these platform organisms have been built, scientists import standardized parts, readily understood sequences of DNA. These well-known sequence code for certain functions, say color or smell. These pieces are stackable or additive, like Legos, so an organism, in theory, could take 2 blocks of DNA to fluoresce green and smell like a banana. The idea is to build microbes and systems capable of performing multi-step processes based on different solutions or different situations. These are simple examples, and like software, the downstream applications are much further reaching. Meanwhile, the design and assembly is done from a computer using specialized software and DNA synthesis. Previously, DNA design and assembly was done by hand, but advances in DNA construction and computer software allow scientists to design their organisms on a computer and get their DNA made to order. Ideally, these functions could be accomplished by machines in an automated laboratory, eliminating human error in the design and assembly process and allowing large numbers of genetic permutations to be tried. The engineering of predictable host organisms, standardized DNA parts, and computer-aided design software has made biology far more deliberate, predictable, reproducible, scalable, and iterative. But the picture I've just painted for you presents the vision for this science, and we are still several breakthroughs away from having truly robust biological systems. Unlike machines, living things have unpredictable processes. They evolve over time, and they evade our complete understanding. Along the path to delivering this new future, there are innumerable tools, assembly methods, standards, and devices that must be built. Many are still in the early stages, and oftentimes these tools have new applications of their own, like CRISPR-Cas9, which is an engineering biology tool that allows for very precise in vivo gene editing, but has applications in many other areas, including the development of new therapeutics. While these systems may be difficult to understand in the abstract, the applications are actually quite powerful in reality. One popular example is the production of artemisinin, an anti-malarial drug. With more than 1 million people dying from malaria each year, most of them under the age of 5, and in sub-Saharan Africa, both access to and the cost of the treatment remains an enormous challenge. Normally, artemisinic acid is produced by the wormwood plant, which has volatile supply and comes almost exclusively from local farms in Vietnam and Thailand that cultivate the plant. One of the leading scientists in synthetic biology, J. Kiesling, engineered a new strain of yeast that creates the chemical precursor needed to produce artemisinin cheaply and predictably. By taking genes from the wormwood plant that code for a series of enzymes and tuning the multi-enzyme pathway for efficiency, Kiesling was able to get the yeast to secrete artemisinic acid, which is then turned into the drug artemisinin with a final chemical step. The Bill and Melinda Gates Foundation put $50 million into scaling up this product and worked in partnership with Sanofi Aventis to produce the drug on a zero-cost, zero-profit model. Sanofi has plans to distribute the drug to 150 million people this year. So artemisinin is a perfect demonstration of the power of this science. And yet, as I mentioned earlier, there are many other applications beyond healthcare, such as in energy, agriculture, bioremediation, and chemicals. Innumerable products from clothing to plastics to perfumes are all made with petroleum-derived chemicals or with specialty chemicals that have environmentally destructive ramifications, such as palm oil. Companies like Ecover are using modified strains of algae to produce palm oil, which will help reduce pressure on the rainforests in Indonesia and elsewhere. Ginkgo Bioworks is working with traditional perfumers to design a cultured rose extract based on numerous molecular attributes that control for fragrance. Other companies, such as Genencor, are working to create a bioisoprene monomer, which will produce high-quality synthetic rubber without dependence on petroleum-based materials that cause adverse environmental consequences. Due to their high price-volume ratio, renewable chemicals are likely to see a large share of the near-term commercial activity in engineering biology. Today, you're seeing price-competitive products derived from sustainable synbiochemicals in creams, flavors, soups, and fragrances. Applications in bioremediation are a topic of popular interest, as organisms can be used to clean up oil spills or consume environmental contaminants such as plastic. Modular Genetics, in partnership with several academic universities, engineered a microorganism that produced soy-based biodispersants for oil pollution management, which could be used to assist cleaning up environmental disasters like the 2010 Deepwater Horizon oil spill in the Gulf, rather than using oils from trees grown in the rainforest. Some scientists are also chasing the idea of reintroducing extinct species as a long-term project by re-engineering nearby relatives or by cloning, replacing the embryonic DNA of a nearby relative with the DNA of the extinct species. Like in Jurassic Park, fiction turns into reality. As long as you have the DNA, the sequence DNA for an extinct species, it is theoretically possible to bring it back to life. And you may ask why we should be concerned with reintroducing the carrier pigeon or the woolly mammoth when so many current species are threatened, with 1 in 6 species facing extinction as a result of climate change since the Industrial Revolution. These efforts may actually help to save species that might otherwise disappear. In agriculture, we're facing multiple challenges, particularly the exponential increase in human population pegged against a linear increase in food production. Some early companies are targeting ways to cultivate a better and safer food system. Clara Foods, for example, has produced egg whites in the laboratory using yeast that does not require the cost and time spent raising chickens. MuFree is working on synthetic milk made from yeast cultures. Modern Meadow is developing cultured food products that do not rely on animals in collaboration with well-known chefs. There are also near-term applications for engineering biology and pest management to control deadly diseases and protect food crops. Dengue fever, a mosquito-borne disease, affects over 50 million people in tropical areas of the globe each year and is estimated to cost the global economy $5 billion per year. There is no medication or vaccine for dengue. The only way to control the disease is to control the mosquito that carries the virus. A company called Oxitec has created modified versions of sterile male mosquitoes which seek out and mate with females to produce offspring that will not survive to adulthood, thereby controlling the mosquito population without pesticides. Recently, the company announced that releases of the genetically modified mosquito in a tropical area of Brazil reduced the dengue mosquito population by 95%, which was well below the threshold for epidemic disease transmission. And finally, renewable energy from non-petroleum-based sources remains the largest promise and to date the most elusive to achieve. Numerous companies such as Amyris and Solazyme raised a lot of money on the promise of leveraging advances in biology that would drive renewable energy. So far, they have had to scale back their efforts because yields on renewable sources like algae or biomass are still not scalable or really price competitive. Nevertheless, Amyris is currently selling renewable diesel in metropolitan areas like Brazil and renewable jet fuel with its partner Total all around the world. Even beyond the applications I just talked about, which are about global sustainability, synthetic biology is also vital for endeavors beyond our planet. NASA, as well as space companies, are using relevant applications in these areas to redesign plants to create ecosystems in space engineer to reduce the effects of radiation and zero gravity in space, detect life on other planets, reduce weight and create new types of fuels, and eventually plans to colonize other planets. But the field is not without its challenges. The first is cost in technological development. As we've just explored, the applications of engineering biology are numerous, and our mastery over writing DNA will continue to increase rapidly. Today, the cost sits at about 20 to 30 cents per base pair, depending on the length of the sequence, but I expect to see another tenfold decrease in the cost of DNA synthesis as companies like Twist Bioscience and Gen9 enter the marketplace. In fact, the rapidity of development is a challenge in itself. New ways of working are straining old methods, and as legal frameworks become outdated quickly, regulatory bodies are slow to adapt, and the public is viscerally uncomfortable with having so much control over the natural world. On the intellectual property side, a tug of war exists between an open source culture that wishes to democratize science and those who understand that patent protection is necessary to commercialize development and the acceleration of scientific development. Consider again the various parts of engineered biology, the host organism and the standardized DNA parts in particular. Many existing standardized parts have been filed under open source licenses that make commercializing products derived from these tools difficult, but necessary if the benefits from them are to be realized. Many of our nation's regulatory bodies are ill-equipped to handle such advances, as are many international bodies. For example, in regard to the release of modified mosquitoes discussed earlier, there were questions about which body should regulate the release of organisms. And ultimately, the FDA approved their release, but the example highlights the fact that agencies lack the expertise to evaluate some of these new applications. Additionally, new applications may span the purview of multiple agencies. In this case, the USDA could have regulated this, or the NIH, or the EPA. The speed of development alone is one of the underlying forces that drive some of the hurdles to this field's smooth development. The regulatory landscape is ill-equipped to adapt to this changing environment, especially considering the expertise that is required to properly understand and evaluate these technologies. Meanwhile, the democratization of science and funding is allowing new entrepreneurs to enter the market. You are looking at a glowing plant, a project based in a community lab in San Francisco that, as their name suggests, wants to make plants that glow in the dark. Their long-term vision is to replace streetlights with glowing trees, thereby reducing CO2 emissions by not requiring electricity. But for now, they'll make small glow-in-the-dark ornamental plants. Glowing Plant funded themselves through Kickstarter, in part by promising their 7,000 funders that they would receive free seeds to grow their own glowing plants as compensation. This sounds fine until you consider the millions of dollars of testing and trials large companies often go through before releasing modified organisms into the environment. These trials are designed to prevent the release of potentially harmful non-native species into the environment. And yet the regulatory infrastructure was designed to work with these large agro-tech companies and not unknown projects in San Francisco. So glowing plants fell through the cracks. Global competition is also a concern because engineering biology is a discipline of science built on a different fundamental premise, designing biological parts and systems from the ground up, and the applications are so far-reaching, the continued development of the field will have a profound impact on the economics of nations who pursue this technology. Being able to engineer the building blocks of life with such control has enormous implications for future leadership and the nature of employment over the next century. One of the risks facing the United States right now is a decline in funding, while competing nations such as the UK and China have pursued long-term, coordinated funding mechanisms aimed at advancing the infrastructure that accelerates fundamental and translational research. England, for example, names synthetic biology as one of the 8 great technologies, along with others such as big data and robotics and animation systems, and has developed interagency funding mechanisms with leading universities and companies for research as well as programs to address ethical, legal, and public concerns. The U.S. risks falling behind economically if it does not dedicate the resources to the advancement of this science. Risks and ethical implications are also an important criteria or concern. Grappling with the implications of such advances can be quite daunting, and this technology raises concerns, both old and new. The old fears have been around as long as science itself, namely safe handling and preventive maleficent use. Since the discovery of recombinant DNA in the 1970s, which essentially marks the beginning of the modern biotech revolution, scientists have generally been at the forefront of addressing issues of safety. Starting with the Asilomar Conference in 1975, for example, scientists convened with regulators to develop safety guidelines and processes for institutional oversight. To date, there have been no accidental releases of genetically engineered microorganisms from laboratories. Today, with engineering biology, scientists have worked to update standards of safety, especially around preventing the synthesis of pathogenic sequences, and in addition, safety precautions are designed within the organisms themselves, as I discussed earlier, with the introduction of safety switches that do not allow the organisms to exist outside of the laboratory. For the foreseeable future, the threats posed are relatively small, as the majority of experiments involve transferring well-known parts into well-known bacterial hosts for use within a lab. While this industry has been generally good about safety, there are a number of modern concerns about DNA ownership, safe release of new organisms, and the public's visceral uncomfortableness with having so much control over the natural world. These questions of ownership will not be resolved today, and will continue to evolve as these questions become more complex. And while there are many open ethical issues, the reality is that the technology itself is ethically neutral. Like almost anything else, it can be used for good or for ill. The onus is really on the scientific community and the public to ensure that the field develops with these considerations in mind. One of the most important synthetic biology techniques to appear is called CRISPR-Cas9, which many of you might have heard of, which allows for gene editing within living cells. The Western world has issued a moratorium on using this science for germline editing, which would make traits heritable. Yet just recently, a team of Chinese researchers, of which there are other teams, submitted a paper in which they tried to edit out a potentially fatal genetic disorder in non-viable embryonic stem cells. The breach of the moratorium brings up a host of ethical issues related to germline editing, such as the possibility for unpredictable outcomes and non-medical enhancements of human beings. As the technology develops, a focus on preemptive ethical discussions will become more and more essential to ensure that these advances are used responsibly. So if this presentation has explained anything, I hope it has shown that advances in engineering biology hold much promise for the world. Engineering biology will, in our lifetimes, have the technological power to remediate many of the world's imminent and most intractable problems. It is not without challenges and risks, however, and improvements in the modern world brought about by technological change often carry unexpected consequences. The challenges of our time still far surpass our capabilities of solving them, and these challenges are not solely technical. If we are to create the world we truly wish to live in, we must work to change culture and consciousness in ways that often cannot be engineered in a lab. Thank you very much. I'd be happy to answer any questions you might have.
Thank you so much, Nancy, for a fascinating and very eloquent presentation on engineering biology. In introducing you, I neglected to tell the audience that you are welcome to submit your own questions, and there's still time for you to do that. We have a good 10 minutes for Q&A here. So you see the small Q&A button to the left of your screen. Please click that and feel free to enter any questions that you may have, and I'll do my best to bring them up and deliver them to get Nancy's response. Nancy, a few questions of my own to hopefully get you to expand on a few of the points that you touched upon in your presentation. First of all, the nomenclature. I spent a lot of time in the introduction talking about your interest and expertise in synthetic biology, and then you tell me it's not called synthetic biology anymore, it's engineering biology. Is this simply a matter of semantics, or is there something more important potentially at play here?
Matter of semantics, and there is much larger issues at play, which is why the field is increasingly turning to the term engineering biology rather than calling it synthetic biology. Originally, when the field was first being developed, the term synthetic biology was used as the term for engineering new biological organisms. But as time went on and the public debate and the need for public education around these issues became more important, it was hard for the public to truly understand how something that was genetically identical, created in the laboratory, that was genetically identical to that found in the real world, could be called synthetic. In fact, they were both identical, and so they were both derived from lessons that we learn in the natural world. And the word synthetic by itself makes people think of things that are artificial. And that's not entirely true in this case. And so I think we're moving towards biologically engineered solutions rather than synthetic biology, although synthetic biology is still being used within the industry.
Okay, thanks very much. That will come as interesting news to the editors of ACS Synthetic Biology. Of course, one of the ACS's leading journals, and one of the leading journals in this particular field. No word yet on if they're planning to change their name. Nancy, my next question is, there may be viewers who are curious as to how you became immersed in this field of engineering biology in the first place. And I know that you spent a good period of time over the past couple of years writing or researching and producing a major report that looked at the future of an alliance that some of the viewers may be familiar with, SynBIRC, one of the leading, perhaps the leading national consortium of synthetic or engineering biology laboratories. I wonder if you could just quickly refresh or inform the audience as to what SynBIRC is, what your mission was, and perhaps what were the fruits and outcomes of that particular piece of research that you conducted on their behalf?
Acronym for the Synthetic Biology Engineering Research Center. It is a virtual consortium of scientists and academic institutions that was created in 2006 as part of the National Science Foundation's Engineering Research Center programs. And it was given approximately $60 million in funding to pursue fundamental research over a period of 10 years. The original goals of the scientists and academic institutions that participated in SynBERC were both audacious and simple at the same time, and they were two. One is that they wanted to prove that engineering concepts could be applied to biology. And secondly, they wanted to build an industry around the science that emerged from that. In the period of just 10 years, they have done that, and we now have a rapidly growing field in which there are many new companies that are emerging and are receiving substantial private sector funding right now. And when I left so SynBERC's funding from the National Science Foundation will sunset in 2016 because that is a 10-year program. And so when I left the New York Genome Center, the Sloan Foundation and SynBERC asked me to take a look at the future of synthetic biology in the United States, and in particular to create a strategic plan for accelerating that field and for perpetuating SynBERC either in its current form or in some other form that we might recommend. And so we spent a year doing a very in-depth analysis of the marketplace. We attended all of the community events. The field had grown from what had started out with about 10 scientists to over 400 scientists across the country that met in a variety of organizations and scientific meetings. We interviewed over 110 people from government, the White House, through the agencies to academic scientists to deans at the academic institutions to industry that is working in this field. All kinds of people. We reviewed over 500 pieces of scientific literature. And one of the biggest things that we found in terms of looking towards sustainability for SynBERC and a strategic plan for the community in general is that no one really understood what had been created from SynBERC. And so they were asking, why should we make additional investment if we don't know what the benefits are that have been created with the first $60 million that was invested? And so the first thing we did was to write probably the most comprehensive report of the field that had been done to date. It's on my website, and a summary of that report was actually published in Industrial Biotechnology in June of 2016. We published the strategic plan in June of 2016, and a commentary by me was also published in Industrial Biotechnology in that same month, calling for a National Center for Engineering Biology that would advance this field, create an interagency funding process, so all the agencies could coordinate and allow companies to continue to work in important applications in this area. So that's the work that I've been doing over the last 2 years. Since we published the strategic plan, we've been working with the broader engineering biology community. Most recently, we co-sponsored some important scientific meetings at the New York Genome Center in Manhattan. One focused on engineering biology and creating a center around that, and the second called SC 2.0, which is the synthetic synthesis of the yeast chromosomes, which is led by Jeff Buca, and it is involved 200 scientists from 8 countries all over the world. So the work is ongoing right now, and we'll continue to have new announcements. I actually have an engineering biology page on my website that is following the progress of all of this work. And it also contains interviews from many of the leading scientists and industry leaders about their thoughts and ideas about why this field is important and what it means for the world.
Thank you. Let's just a couple of questions, I think, before we wrap up here. Given Nancy, as I said in the introduction, you did something absolutely remarkable 2 or 3 years ago, not just in conceiving, but seeing this the physical creation of a world-class genome center right in the heart of Manhattan. With all of the leading New York academic institutions and others further afield participating both financially and intellectually. Do you see down the road potentially any sort of similar institute taking shape in the field of engineering biology?
Yes, we actually held a meeting in April that was co-sponsored with the Sloan Foundation and the Woodrow Wilson Institute. We had 60 people from the scientific community and industry discussing the need for such a center, and I think the overwhelming consensus was there was the need. The meeting summary, again, can be found on the engineering biology page of my website. We had a follow-on meeting in July, as I just described. And one of the things that has come out of all of this discussion is that many of the scientists who are not necessarily associated with an academic institution, who may be citizen scientists, who may be independent scientists, want to have access to these facilities so that they are able to use the tools of engineering biology to create their own products and perhaps build companies around them. And we could imagine an open source infrastructure that could be accessed from scientists all over the world that would allow access to these design, engineering, and synthesis tools, as well as data storage and ways to share information using platforms like the one that has been designed by Stefan Friend at SAGE. So there's a lot of potential in this area, and I think one of the things that will move it forward most rapidly is the definition of a grand challenge in this field where scientists could actually begin to work collectively and attract collective funding either from the philanthropic, the private, or the public sectors.
I just want to segue from that response and ask the final question before we close. We're just a few hours from the next Republican debate, which should be must-see television. I wonder how optimistic you are, or if you see any signs of encouragement about the future of not just science funding in general, but funding for engineering biology in particular. What would you like to see coming out of this upcoming debate and amongst all of the candidates of any party over the next 12 months?
I think that there are some promising signs. Obviously, this is quite a controversial field, and there is widespread political positions across the spectrum. There are some encouraging developments. So a bipartisan bill on engineering biology in 2015 was introduced into the Congress in January of this year. It is not an appropriations bill, so it does not have funding attached to it, but it would create a federal program focused on engineering biology, supported by an interagency funding process. And it would have a lot of components such as public engagement, public education that is really required in this area. There's also been some movement in the White House recently to create more transparency in regulation and a call for the agencies to actually have some interactive discussions about rules and regulations and the approaches that they will take to regulating this kind of new biology. And the EPA is having its meeting on that call just shortly. So there are some promising things that are starting to develop. I don't have a crystal ball about what's actually going to happen.
Well, Nancy Kelley, the founder and principal of Nancy J. Kelley and Associates, and a leading authority and expert in engineering biology and life science funding. Thank you so much for a wonderful presentation and a great way to round out the 2015 CNEN Virtual Symposium. So thanks to Nancy and the dozen other speakers who generously gave of their time and delivered us a series of fascinating and cutting-edge talks over the course of the whole day today. I particularly want to thank my colleagues at CNEN and LabRoots partners here for working so hard behind the scenes to prepare and put this great show on. We like this virtual environment very much. And in particular, thanks to you for watching and the thousands of other scientists and colleagues who tuned in at various points during the day to take advantage of this. All of the talks will be available on demand through the rest of the year, through the end of 2015. So I hope you'll share the news with your colleagues and encourage them to come back and view those that you may have missed. We'll be back in 2016, I can assure you, with some more virtual events on important, timely subjects of particular interest to the chemistry community, so stay tuned for news of those. For now, and for CNEN, my name is Kevin Davis. Thank you so much again for watching, and goodbye for now.